Nucleic acids and proteins. Nucleic acids. There are two types of nucleic acids in all cells. One, DNA, or deoxyribonucleic acid, which stores information. And two, RNA, or ribonucleic acid, which carries information from DNA to other structures in the cell. In eukaryotic cells, these molecules are found in the nucleus. Unlike DNA, RNA is also found in the cytoplasm. Prokaryotic cells, however, lack a defined nuclei, and DNA is found in an area called the nucleoid. DNA function. DNA stores all the information that cells and organisms need for life. It is known as the blueprint of life, or genetic code, or molecule of heredity. Genetic information flows from DNA in two ways. Chromosomes store the information that is inherited from one generation to the next, and carry all of the genetic information for an organism. Genes, short segments of DNA, store encoded RNA and protein. Genes provide all the information that an organism needs to grow, develop, and carry out all the daily functions of life. DNA and chromosomes. All cells, both prokaryotic and eukaryotic, store their hereditary information in chromosomes, using the same nucleotides. The number, types, and the order of the nucleotides in each organism encodes unique genetic information. Chromosomes are made of DNA wrapped around protein scaffolding proteins. Prokaryotes usually have one large circular chromosome. Eukaryotes usually have a number of linear chromosomes, sometimes a very big number. A bacterial or archaeal chromosome is circular, as seen in red, and the eukaryotic chromosome is linear, as seen in blue. Nucleotides. Nucleic acids, the polymers, are composed of chains of nucleotides, the monomers. Nucleotides are complex monomers. They are made of three subunits: a sugar, a nitrogenous base, and a phosphate. One molecule of DNA can be made of more than 10 to the 8th nucleotides. Bonds in nucleic acid, DNA. DNA is double-stranded. Each chain is formed by strong covalent bonds between the sugars and phosphates together, forming the sugar-phosphate backbone. The nitrogenous bases on the chain bond to the bases on the complementary chain with weaker hydrogen bonds. The weaker hydrogen bonds allow short segments of the DNA molecule to be split or melted apart, copied, and then put back together. Sequence of nitrogenous bases carries information. This diagram shows nucleotides of a strand of DNA in detail. The backbone of the molecule repeats alternating sugar-phosphate, while the nitrogenous bases protrude from the backbone. It is the varying sequence of these bases in RNA and DNA that stores coding information. Complementary base pairing stabilizes DNA. The aggregate force of millions of hydrogen bonds in a chromosome help to create a very stable, double-stranded structure. The nitrogen bases hydrogen bond with their complementary bases on the opposite strand, A with T and C with G. RNA versus DNA. While RNA and DNA are both polynucleotides, they differ in structure and function. This diagram shows the RNA's nitrogenous bases include uracil, while DNA substitutes diamine for uracil. RNA and DNA are further compared on the next slides. RNA versus DNA. RNA is located in the nucleus and cytoplasm of eukaryotes, while DNA is just located in the nucleus. The functions of RNA vary. It could be in the form of mRNA, which is messenger RNA, carrying information from genes to ribosomes. rRNA, or ribosomal RNA, forms the structure of ribosomes. tRNA, or transfer RNA, translates mRNA sequences to amino acid sequences. The functions of DNA include storing inherited information in the form of chromosomes, and DNA contain genes, which are short segments of the chromosomes that encode RNA and proteins. RNA versus DNA. RNA has single strands with secondary structure, forming shapes, and DNA is double-stranded. RNA contains the sugar ribose, while DNA contains the sugar deoxyribose. The base pairing in RNA, uracil U pairs with adenine A, and cytosine C pairs with guanine G. In DNA, thymine pairs with adenine A, and cytosine C pairs with guanine G. RNA. RNA molecules are made of single strands of nucleotides that fold in on themselves, forming hydrogen bonds between adenine and uracil, or cytosine and guanine, giving them secondary structure. As we saw in proteins, differences in primary structure lead to differences in higher order structures, differences in shape, and structures and function. A different sequences of bases leads to different shapes and function.